Respected audience, Assalamu alaikum. I am Professor Shamsuzzaman, Professor of Pathology. I welcome all in today's 43rd lecture on pathology. Today, day 9 of Neoplasm. Dear audience, today's topic is biology of tumor growth and tumorigenesis. Biology of tumor growth, tumorigenesis. First come to biology of tumor growth. Dear audience, what we mean about biology of tumor growth? Biology of tumor growth means natural history of tumor. So it is the natural history of tumor. Natural history of most of the malignant tumor natural history of most of the malignant tumors are malignant change in target cell. Malignant change in target cell. It is known as initiation. It is known as initiation. And when there is malignant change in target cell, this cell is known as initiated cell or transformed cell. So, the target cell with malignant change is known as initiated cell or transformed cell. Target cell with malignant change is known as initiated cell or transformed cell. Dear audience, suppose this is liver. If within the liver, this is the target cell. This is the target cell. So if there is malignant change within this target cell, then the target cell with the malignant change is known as initiated cell or transformed cell. First one is malignant change in target cell. Second one, growth of initiated cell. Growth of initiated cell. That is, the initiated cell undergo multiplication. So, the cell undergo multiplication. So growth of initiated cell the initiated cell undergo multiplication. Again, the target cell with the malignant change is known as initiated cell. So this is initiated cell. Third one is local invasion of tumor. Dear audience, the target cell with malignant change is known as initiated cell. Initiated cell undergo growth and multiplication, formation of a tumor. The third one is local invasion of the tumor cell. The tumor cell invades the tissue of this organ. This is local invasion of the tumor. The fourth one is distant metastasis. Distant metastasis. So the target cell undergo malignant change and it is known as initiated cell. Initiated cell multiplies, the growth of initiated cell and formation of tumor. The tumor cell local invasion, and this tumor cell or malignant cell goes to another organ like lung and makes a second tumor. This is known as metastasis. This is natural history of most of the malignant tumors. This is the natural history of tumor, and this is biology of tumor growth. It includes malignant change in target cell, growth of initiated cell, local invasion, and distant metastasis. Dear audience, I have told you here natural history of most of the malignant tumors. We know there are some malignant tumors, they are locally malignant tumors, they don't metastasize. If the malignant tumor does not metastasize, there is no distant metastasis in that malignant tumor. So I have told you this is natural history of most of the malignant tumors. Dear audience, now come to tumorigenesis. It is also known as oncogenesis or carcinogenesis. So, tumorigenesis, that is oncogenesis, that is carcinogenesis. Dear audience, the molecular basis of carcinogenesis, molecular basis of tumorigenesis, molecular basis of oncogenesis is genetic mutation. Genetic mutation is the molecular basis of tumorigenesis. That is, genetic mutation is the molecular basis of carcinogenesis, is the molecular basis of oncogenesis. And genetic mutation occurs in the germline or in the somatic cell. Genetic mutation occurs in germline and also in somatic cell. Dear audience, genetic mutation is the molecular basis of tumorigenesis, and this genetic mutation occurs either in the germline or in the somatic cells. Now come to what are the genes undergo mutation for carcinogenesis. genes undergo mutation in carcinogenesis, in tumorigenesis, in oncogenesis. What are the genes? One is Proto-oncogene, another is Tumor suppressor gene, Proto-oncogene, tumor suppressor gene, gene regulating apoptosis, gene regulating apoptosis. Gene required for repair of damaged DNA. Gene required for repair of damaged DNA. So these are the genes undergo mutation in carcinogenesis, proto-oncogene, tumor suppressor gene, gene regulating apoptosis, gene required for repair of damaged DNA. Dear audience, we know elimination of unwanted host cell through a series of events programmed by a dedicated set of genes is known as apoptosis. If a cell becomes unwanted in our body, is a host cell, this unwanted cell is eliminated by the process of apoptosis. If there is mutation of the gene regulating apoptosis, the unwanted cell will not be eliminated from our body, and this unwanted cell may produce neoplasm. In our body, if there is damage of DNA, the damaged DNA is repaired, and this repair of damaged DNA is controlled by gene. If that gene undergo mutation, the damaged DNA will not be repaired, and there is chance of mutation of that DNA. Now come to Proto-oncogene. What is Proto-oncogene? Proto-oncogene is present in our cell, and this gene is associated with normal cell division and differentiation. Gene associated with normal cell division and differentiation is known as Proto-oncogene. Dear audience, this gene is associated with normal cell division and differentiation. Suppose this is reserved cell. This is reserved cell in an organ or in tissue. The reserved cell is undifferentiated cell. If this cell undergo multiplications, and this multiplications is due to proto-oncogene, and this is the undifferentiated cell. After multiplication, this cell may differentiate to epithelial cell, or it may differentiate to connective tissue cell. Whether the reserved cell will multiply or not, and after multiplication whether this cell will differentiate to epithelial lineage or to mesenchymal lineage, it depends upon the proto-oncogene. So proto-oncogene is associated with normal cell division and differentiation. Tumor suppressor gene. Tumor suppressor gene, it is present in our body. This gene is associated with regulation of normal cell division. Gene is associated with regulation of normal cell growth is known as tumor suppressor gene. So the gene associated with regulation of normal cell growth or normal cell division, it is known as tumor suppressor gene. In other words, this gene controls and suppresses. In other words, this gene controls and suppresses the overactivity of Protooncogene. So gene associated with regulation of normal cell growth or normal cell division is known as suppressor gene. In other words, this gene controls and suppresses the overactivity of Protooncogene. Thereby, it reduces, this gene reduces, risk of tumorigenesis. So this gene reduces the risk of tumorigenesis. Dear audience, suppose this is the reserved cell and due to Protooncogene, this cell undergo multiplication. The tumor suppressor gene controls the Protooncogene and suppresses its overactivity. Dear audience, suppose this is reserved cell and Protooncogene is associated with the multiplication of this cell and there is formation of new cells. This Protooncogene is controlled by tumor suppressor gene. If tumor suppressor gene fails to control the Protooncogene, there will be uncontrolled proliferation. Again, if the tumor suppressor gene fails to control the Protooncogene, there will be uncontrolled proliferation of cells. Uncontrolled proliferation of cells. And due to uncontrolled proliferation of cell, there is formation of tumor. Again, if tumor suppressor gene fails to control the Protooncogene will be uncontrolled, and due to uncontrolled proliferation of the cells, there will be formation of tumor. This Protooncogene is controlled by tumor suppressor gene. The overactivity of the proto-oncogene is suppressed by tumor suppressor gene. So there is no tumorigenesis and there is reduction of risk of tumorigenesis. Now come to some name of tumor suppressor gene. RB gene, retinoblastoma gene. Mutation of RB gene is associated with tumor retinoblastoma. It is a childhood malignant tumor. Mutation of RB gene is associated with retinoblastoma. Mutation of RB gene is associated with retinoblastoma. P53 gene, non-mutated P53 gene reduces risk of tumor. When there is mutation of P53 gene, there will be tumorigenesis. Mutation of P53 gene is associated with tumorigenesis. Dear audience, P53 gene is also known as molecular policeman, also known as guardian gene, also known as wild type gene. This gene reduces the risk of tumorigenesis. So it is known as molecular policeman, it is known as guardian gene, and is also known as wild type gene because this gene reduces risk of tumorigenesis. Dear audience, now come to oncogene. The gene associated with oncogenesis, the gene associated with tumorigenesis, gene associated with carcinogenesis is known as oncogene. So oncogene is not present in our cell, rather proto-oncogene is transformed to oncogene. So the gene associated with tumorigenesis, gene associated with carcinogenesis, gene associated with oncogenesis is known as oncogene. It is not present in our body or in cell. Proto-oncogene that is present in our cell is transformed to oncogene. it is not present in our cell, but Proto-Oncogene is transformed to Oncogene. Now come to how Proto-Oncogene is transformed to Oncogene. Dear audience, I have told you genetic mutation is the molecular basis of tumorigenesis. Until and unless there is genetic mutation, there will be no tumorigenesis. When Proto-Oncogene is transformed to Oncogene, there is chance of oncogenesis. Now come to how Proto-Oncogene is transformed to Oncogene. How Proto-Oncogene is transformed to Oncogene? Karyotypic changes is associated with transformation of Proto-Oncogene to Oncogene. Karyotypic changes is associated with transformation of Proto-Oncogene to Oncogene. So, karyotypic changes is associated with transformation of Proto-Oncogene to Oncogene. What are the karyotypic changes? The karyotypic changes that are associated with transformation of Proto-Oncogene to Oncogene are balanced translocation, dilation, and gene amplification. So karyotypic changes are balanced translocation, dilation, and gene amplification. Dear audience, now come to balanced translocation. What we mean about balanced translocation? Before going to discuss this, we have to recapitulate. Suppose this is a chromosome with short arm, and this is the long arm, and this is the centromere. I have told you about the Proto-Oncogene associated with normal cell division and differentiation, and tumor suppressor gene that controls the Proto-Oncogene. Suppose this is the tumor suppressor gene, and this is the Proto-Oncogene. So this tumor suppressor gene suppresses the overactivity of the Proto-Oncogene, and so there is no tumorigenesis. What happens in balanced translocation? In balanced translocation, between two chromosomes, a segment of chromosome is exchanged. Between two chromosomes, a segment of chromosome of same length, of same arm is exchanged. Again, between two chromosomes, a segment of chromosome of same length, of same arm is exchanged. So it is called balanced translocation. Suppose this is a chromosome This is centromere and this is the short arm. This is another chromosome. This is centromere and this is the short arm. Suppose between the short arms of these two chromosomes, between the short arms of this chromosome, same length is exchanged. After exchanging of this length, we will get the chromosome like this. So this is the balanced translocation. Dear friends, again I have told you the tumor suppressor gene controls the proto-oncogene. Dear friends, during balanced translocation, if the proto-oncogene is displaced to a place where there is no tumor suppressor gene, then the proto-oncogene, without having the control of the suppressor gene, become uncontrolled, and so there will be uncontrolled proliferation and tumorigenesis. Again, if the proto-oncogene is placed to a strong promoter, then the proto-oncogene will be uncontrolled, and there will be uncontrolled proliferation and tumorigenesis. Dear audience, now come to dilation. Suppose this is a chromosome, this is centromere, and this is the proto-oncogene, and this is the tumor suppressor gene. Dear audience, in deletion, a segment of chromosome is deleted forever. If the deleted part takes the suppressor gene, then the proto-oncogene become alone. Suppose this part is deleted. After deletion, the chromosome becomes like this. This is the centromere and this is the proto-oncogene. So after deletion, this is the deleted part bearing the tumor suppressor gene. So in deletion, if the deleted part is deleted, taking the tumor suppressor gene and leaving the proto-oncogene, in that case the proto-oncogene become alone. Now this proto-oncogene is alone. When the proto-oncogene become alone, there is no control of tumor suppressor gene, so it undergoes uncontrolled, and there is uncontrolled proliferation and tumorigenesis. Now come to gene amplification. Suppose this is the chromosome, centromere, this is proto-oncogene, this is tumor suppressor gene. In gene amplification, there is no deletion, there is no balanced translocation. Rather, the proto-oncogene undergo amplification. The activities of the proto-oncogene is amplified due to exposure to certain carcinogen. Due to exposure to certain carcinogen, the activities of the proto-oncogene is amplified without translocation, without deletion. And as the activities of proto-oncogene is amplified, there will be uncontrolled proliferation and tumorigenesis. So these are the karyotypic changes associated with transformation of proto-oncogene to oncogene. Dear audience, when the proto-oncogene become uncontrolled without the control of the suppressor gene, without of the suppression of the tumor suppressor gene, this is known as transformation of proto-oncogene to oncogene. So these are the karyotypic changes, balance translocation, deletion and gene amplification associated with transformation of proto-oncogene to oncogene. Now come to steps of tumorigenesis. I mean steps of carcinogenesis, steps of oncogenesis. There are two steps. One is initiation, another is promotion. First come to initiation. What happens in initiation? In initiation, due to exposure of cell to certain carcinogen, there is genetic mutation. What happens? Genetic mutation due to exposure to certain carcinogen. So genetic mutation occurs due to exposure to certain carcinogen in initiation. And after the genetic mutation within the cell is known as initiated cell. After having genetic mutation, the cell is called initiated cell or transformed cell, called transformed cell. Dear audience, if this is a cell, nucleus, chromosome, and if this is the carcinogen, appropriate carcinogen, there will be genetic mutation. Genetic mutation in cell. And with the genetic mutation, the cell is known as, this is known as initiated cell. This is initiated cell or transformed cell. Now come to what are the characteristics of initiated cell. Characteristics of initiated cell. Number one, the cell has no growth autonomy. The initiated cell has no growth autonomy. Dear audience, there is genetic mutation within the cell and this cell is known as initiated cell. In the first step, there is formation of initiated cell. The initiated cell has no growth autonomy. That is, the cell is not capable alone to grow a tumor. The cell is not capable alone to grow a tumor. Number 2, cell has no genotypic or phenotypic marker. The initiated cell has no genotypic or phenotypic marker for ready identification. Cell has no genotypic or phenotypic marker for ready identification. Dear audience, on exposure to carcinogen, there is development of initiated cell in our body, but we cannot identify that cell because the cell has no genotypic marker or phenotypic marker. Another characteristics, initiation is not enough for tumorigenesis. That is, genetic mutation is not enough for tumorigenesis. Initiation is not enough for tumorigenesis. Another characteristics, initiated cell must undergo at least one cycle multiplication. Initiated cell must undergo at least one cycle multiplication to fix the change or to become permanent. Suppose this is normal cell and on exposure to carcinogen, the cell become initiated with genetic mutation. This cell alone is not capable to produce growth. This cell has no genotypic or phenotypic marker. This cell must undergo at least one cycle multiplication so that the change become fixed, so that the cell become permanent. These are the characteristics of initiated cell. Dear audience, on exposure to carcinogen, initiated cell is formed in our body. Now come to the fate of outcomes of initiated cell. Fate of initiated cell. If this is the initiated cell, what may be the fate? Dear audience, you know initiated cell is an unwanted cell in our body. And you know unwanted cell is eliminated from our body by the process of apoptosis. So, the important fate of initiated cell is degeneration. Degeneration of initiated cell. That is, the cell is eliminated by the process of apoptosis as it is unwanted cell. Another fatal outcome, it may progress. It may progress and produce tumor. So these are the fate of initiated cell. Now come to promotion. I have told you two steps of tumorigenesis. One is initiation, another is promotion. Now come to promotion. Dear audience, several substances are associated with promotion. like phenol, hormone, drugs like phenobarbital. Dear audience, these are associated with the promotion. What is the role of such promoters? What are the role of these promoters? Dear audience, in the initiated cell there is genetic mutation, you know. This promoter cannot make change in the gene or DNA, but this promoter can sense the expression of the gene, and without change of the gene but change in the expression of gene is known as epigenetics. So by epigenetic process the promoter changes the expression of the gene, and so there is tumorigenesis. What is the role? Promoters do not sense the DNA, rather it changes the expression of DNA or gene and causes tumorigenesis. The promoters changes the expression of the mutant gene, and so there is tumorigenesis. This is all about the tumorigenesis. Dear audience, today I have told you about the biology of tumor growth. This is the natural history of most of the malignant tumors. There is malignant change in the target cell. There will be growth of the initiated cell. There is local invasion of the tumor and distant metastasis of the tumor. I have told you about the tumorigenesis. There are two steps. One is initiation and another is promotion. In initiation, there is genetic mutation occurs, and in promotion, several substances are associated with the promotion like phenol, hormone, drugs. This does not cause any change in the DNA, but this causes changes in expression of the gene or DNA, and so there is tumorigenesis. Dear audience, today up to this, thanks all.